thank you for the organizer for inviting me. So my lab is here in LSEC, and we're interested in imaging of the human brain with MRI and into developing biophysical models of this measurement in order to actually try to assess changes in development and in aging. I will talk today about aging. Okay. So this is a typical MRI image of an anatomical scan of a brain, and you can appreciate the fact that we can see the gray matter, the white matter, subcortical region, ventricles, and so on. So there is information about really grayscale organization, at least in the brain. And if you would scan, say, a patient with Alzheimer, it would look something like that. So we'll see a huge reduction in brain volume and atrophy. And that will mimic at least what we know in terms of post-mortem in these great changes in brain organization, also in Alzheimer. But MRI in general, or this type of MRI, this is not very specific. And in terms of, if we would look on brain structure in this scale, and we can measure these changes, say, in cortical thickness as function of age, and we'll find actually shrink it with age. So in this case, Alzheimer's is just an extreme case of aging. So that's a perspective you might get with MRI. And so that's kind of idea of something is falling apart. At least some type of atrophy. But if we would look on literature from animal work, invasive work, we'll find like, and we hear a bit about that already, we'll find like a huge amount of information about the molecular changes that happen in cells during normal aging and during diseases which relate to aging. So there will be a lot of mechanism of cellular mechanism that will change the, if you want, the proteomics and lipidomics of cells due to aging, right? And so while in MRI we see something falling apart in very general, there's actually a lot to say about what is changing in this complex biological system, which we're missing right now with MRI. And in my talk, I will try to maybe start and try to close this gap and try to at least retrieve as much information as we can from the MRI about this molecular composition and changes in molecular composition. Okay, so the outline is something like that. I will try to argue for this idea of in vivo histology with MRI, and then show you an approach to do a new approach that we developed in the lab to do that. Show you how we apply that for the human brain, and then for the case of aging, and in the end, if I would have time, maybe also about some theories about aging and how that looks with this new approach. Okay, so let's start with this argument of in vivo histology. So one thing we cannot change is that MRI is measuring in very low resolution, right? When we're really good about that, it's maybe sub-millimeter, but typically around a millimeter resolution. So that's low resolution for this molecular variability. And the idea in vivo histology, or this thought, is that maybe we can find some kind of a way to close the gap and do essentially a biophysical model that will try to relate whatever we measure in MRI to the cellular composition. And in order to do that, the first push and the excitement of this for this idea came from the idea that we actually don't have just to measure images. We actually can measure maps with physical units. So if you have physical units, maybe you can derive a biophysical model of what actually happened to the tissue. So what do I mean by quantitative imaging with physical units? Here is an example of an array of measurements. So you can see that now we see, again, images of the brain, but now they have units, different units. For instance, relaxation, so it's like 1 over time. And so we have measurement with units which are reliable and understood in terms of, at least of physics. And so then the next step was just to ask, what are they relating to? And people were excited because they discovered that, that if you compare these quantitative measurements, say T1, one of them, so it's a relaxation time, to histological work, to histological measurement of myelin, for instance, they found a very strong agreement between T1 and relaxation. So okay, so the assumption is T1 is myelin. And then there's an array of measurement, and they all claim to be myelin, in a way. And so, so this is work that compare different state of myelinization in rodents, and they compare two other methods of quantitative imaging, and they all say, so one of them is T2-based, and the other one is magnetization transfer-based. I'm not going to teach MR physics now. And they all were in great agreement to, or some agreement to, so the myelin estimation from the MRI and the histological estimation of myelin was in good agreement. And then, so they all, there's a little bit of competition. Who is myelin? And then this is a work that came from our lab, from Shai. And she said, okay, you know, we're actually measuring water in MRI. Let's just check if we're just going to measure water in MRI. Would that look like myelin? And the answer is yes. So she took the same data, actually, and she just put here 1 minus water fraction that we can estimate from MRI. That was also in kind of good agreement with histology, actually as good. And so the argument is that water is actually, well, you can say that water is a proxy for myelin. That's one option, or non-water. The other thing is that MRI is very sensitive to water, and that's all correlate to myelin. And if we want to say something more, we need to find a way to tease these things apart. And that's what I'm going to try to show you. So if we put here's the thing. So here we have a measurement of one of these quantitative measurement between two groups. And there's an age difference, and we can ask, is it because of myelin? Is it because of water? Maybe something about the molecular microenvironment. That's kind of the question. And so we want to take this MRI, this anatomical image, and really think about it as a big area of fat and water, actually. That's my perspective of the brain. And try to be able to be sensitive to this interaction of water, which is MRI sensitive to, and then be able to say something about the composition of macromolecules, lipids, and so on. And so the problem is that we have water that is a great effector of our measurement, and we need to remove it somehow or control for it somehow, and then maybe we'll be able to say something about the composition. Okay. So the first thing we need to establish that we have a good measurement of water. And that goes with a previous work that I was doing still at Stanford, in which we show that we can measure this non-water fraction, or what we called in, just give it a good name, MTV, the macromolecule tissue volume, lipid and macromolecule tissue volume. So we have a measurement like that. It has units. And recently, we actually validate that it's actually true. We validated before, but now we have another validation. I will show you a few. That is a true measurement of fraction of water. This is put just vial of water and D2O. D2O we don't see in MRI. Water we do see, and we can actually, so we know exactly what is the fraction of water, and we can measure it with high accuracy. That's what we're seeing here. And then we also did it with animals. This is taking an animal, scan it, and then estimate the water post-mortemly, and we find a very good agreement between the MRI and the non-MRI invasive measurement of water fraction. Okay. And so we have this measurement of water, and there is also, if we look back into the literature, and that's what we did, to the 80s and 90s, people actually did NMR work, so not MRI, but related. And they say, you know, lipids actually important for the MRI signal. So we ask ourselves, can we also, so essentially we went back to this work, tried to do it in MRI, and then tried to bring it to the human brain. So that's what I'm going to show now. And so that's start with the work of Ostrat in our lab. We actually took phospholipids, common phospholipids, sorry. Jump to the figure here. Common phospholipids, create these bubbles of phospholipids, so called liposomes. And she really controlled the type of phospholipid and the concentration of the phospholipids. So she created those envelope of phospholipids in water, and that's how they look in EM, and put them inside vials into the scanner and scan them. So just as the same scan as we do for humans, and we have quantitative units for that. And so what we find is that if we just plot the quantitative measurement, whatever it is, and I'll show you a few, to the non-water fraction, we find this, this trend, this linear trend. Essentially, water is important. We find a linear relationship between the quantitative measurement and the water or non-water fraction. Okay. But the interesting part, and that's where it starts to be interesting, is that when she's changed the type of phospholipids, we always get a linear relationship. And I'll showing you just 3 examples, but we have more. But the relationship changed. So if you want the slope of these linear curves change between the type of phospholipids. And so, and that's kind of given us a unique way to identify different phospholipids in this very, very controlled system, at least. And we kind of term this derivative of the quantitative measurement to the non-water fraction as the relaxivity of the tissue. Here, the relaxivity of the phospholipid. And when we change the type of measurement, we got, again, linear relationship, but different. So the argument here is that we can take many quantitative measurements, relate them all to the water fraction, or account for the water fraction if you want. So looking on those slopes. And we will get this kind of spider plot, which represent the effect of the water on this relaxation measurement per lipid. This is for sphingomyelin, for instance, but then we can do it for others. And we're going to get different spider plots, and in general, you can see that there is information of the phospholipid using this combination of measurements in this way. Okay. One interesting thing that was everything was very pure. The next step was, can we do it with mixtures? That would be the next move, and then we're going to move to the brain. So with mixtures, there was this question, if we know something about this relaxivity or linear relationship of a pure lipid, what will happen if we make a mixture? And the answer, or we ask if it's just summed as linear systems, so it's just a linear summation. That would be very useful if that will be the case, right? We always like linear systems. And so we put the linear measurement and try to predict a mixture of phospholipids. And the answer was that it's actually pretty good. We can make a good prediction of a mixture from a simple, from a pure phospholipids. That's good. And that also allows you to take this equation and rewrite it, right? And try to predict then the fraction of each phospholipid in a mixture. And so we did, and in many cases, but not in all cases, we can actually predict very well the fraction of phospholipids in our what we call phantom system, in this in this experiment, for instance, for phosphocholine, for sphingomyelin, not so much for phosphodiecholine. So this is the most common phospholipid in the brain, or in any cell, actually. Okay, so now, and maybe you got the feeling that there's something about the molecular composition that we can retrieve from MRI if we account for water and other relaxation. That's what I tried to say here. And now we have to ask ourselves, does it work in the brain in any way? That's informative in any way. And so, yeah, now we're going to look at the brain. And again, remember, the brain is much more complicated, right? There's many, many, many phospholipids and proteins and sugars and so on. And so we have to be, but we want to try to see if we have some kind of a signature of the tissue here, at least. And then, so here was measuring a subject with different kind of quantitative measurement and with a measurement of water fraction, and then segment the brain to different regions and ask what happens in those regions. Can she find the same kind of trend as we saw in the phantoms? And here's an example of one subject, for instance. So she take here a piece of the brain. This is the pallidum, so she segment the pallidum, okay? And then she just plot the fraction of non-water against a relaxation measurement, okay? And we find this linear trend. And so at least it's linear here. And adding other areas, this is the frontal white matter. Same thing, it's linear, but in a different slope. And this is the thalamus. Again, linearity, but with different slope. And if we add the other quantitative measurement, in this case, actually pallidum and thalamus have the same slope, but the white matter have a different one. in general, the idea of having multiple quantitative measurements seems to be useful. This is across subject. Okay, so what we find here that first there is different signature for different regions. Okay, and those signatures are varying across region and across measurement, and they are reliable across subject. Okay, so in a way, this is exciting because it's a new way of retrieving information from MRI. And so I can plot again this spider plot, and we're going to get beautiful images, which is the signatures of what we will claim the molecular composition of this brain region using MRI. And now I need to convince you that this is actually related to molecular composition in the brain. So we did a few things in order to gain this information or to check this information. So we have these signatures, which are 4D, right? And we try to look for works, histological work, that actually measure multiple lipids or protein across brain regions. There's actually not so many, surprisingly, particularly for lipids. In the human brain, we found this work from the actually pretty old work that was doing lipidomics. And there they measured 5 type of the main phospholipid in the brain, and then other 2 molecular measurement of proteins to lipids and cholesterol as well. So we have 7 measurements. They measured in 7 regions. So that's what we have from histology. And then we have our 4 measurement. Again, so we're going to look again on the same 7 regions. So this is a little bit of not enough information to do the full comparison. So what we did is, so this is the regions. We did a PCA. We just look on the main variability in terms of molecular signature and come from histology versus our signatures. And we're going to look on the projection of those PCAs between the 2 type of measurement. And we got a remarkable result, actually. We found a really nice agreement between the in vivo measurement to the ex vivo, to the molecular measurement. Okay, so it seems that our signature really captures something about this molecular composition across brain regions. And we can do the same move we did before. We can write it now as a linear equation, right? And try to predict the fraction. And in some cases, we actually can predict from those data, at least. So it's predicting in the sense of cross-validation. So you fit on some and check on the other. And so for sphingomyelin and for the ratio between lipid to proteins, we had actually a nice prediction, but not for all the measurements. So some of them we didn't. OK. So now the next step was to ask, can we do it on the same brain? Because that was like histological work from the 90s, our data. So we went and scanned a porcine, so pigs. And that was and then we dissect this brain. So we did the scanning, then dissect it, and then sent it to lipidomics. And that was done with the help of Adi call from the Goshen lab. So the first thing was to check if we find this linear relationship, and this is just an example of 3 regions, and we do. So the same thing, also not surprising, also in the post-mortem we find the same kind of signatures. And then we add lipidomics works, kind of separating the different type of phospholipids in the tissue and relate, again, with the same approach, with the PCA, the signatures to the signatures of the histology. And we find a remarkable agreement, or a pretty strong agreement. I don't know if it's remarkable anymore. We find a strong agreement between the measurement. Actually, when we remove some of the outliers from the histology, it gets better. And the interesting thing was that if we just compare the water fraction to that or the core quantity measurement, there was no agreement between the histology and the in vivo or ex vivo MRI scans. So we really think that this manipulation of removing the water effect is important in order to assess, in this case, the lipid composition or the variability in lipid composition across the brain here. OK. We also ask ourselves, can we relate that to other histological or post-mortem molecular markers? In this case, in collaboration with Sagiv and Noga that was working in our lab, we went and looked and tried to compare between gene expression in the Allen Brain Institute to our measurement. So what Sagiv and others are actually doing is to take this sea of information from gene expression and try to look of genes that co-vary together across the brain. And then they identify families of genes that co-varied in sprains in their expression. And so we can find about 19 modules of families of genes that co-vary together along the cortex. And when we try to relate those families of genes to our measurement, we find that there were two that were really strikingly and strongly related to our measurement, and that was the family that was related to membranes, and the one which was related to synapse. Okay, so it seems, again, so it's another, if you want another validation of the relevance of this measurement, it seems that genes that are related to, and that's very general, right? The way they give names to these families is interesting by itself. But membrane and synapse families were related to our measurement. Okay. Yes. So now aging. So we have these signatures across brain regions. Do they vary as function of age? Is this an interesting information at all? Because we know from post-mortem, or from, for post-mortem that there's supposed to be a molecular change, right, as function of age. So that was the question here. Can we find a change in these markers? And so if you remember, I was posing this question, what is the source? Is water? Is it microstructure? Maybe something else. At the beginning, I was talking about atrophy. What is actually changing? So I will give you just 3 examples just to give you a sense of the data, I guess. So here is a case in which we measure, looking on one of the quantitative measurement between 2 groups, a young group and an older group, and we don't find a change in the quantitative measurement. But when we breaking it to water fraction and the signature of macromolecules, we actually find a change in both. So that's an example. And if we will just look on the signatures of these signatures, we actually find this is frontal cortex. So in color is the younger group, and in gray is the older group. And we find actually remarkable changes in these molecular signatures in these regions, in this region. When we move to, so here is a case when we do find a change in the quantitative measurement. This is for the cowlet. And then in this case, we actually don't find a change in water, but only in the signatures. Okay, so we actually, it's different than what I showed you before. So different regions actually show different pattern in terms of aging. And this is the signature in general. It's a great change in one of them more than the other. And just a third example, just to sense the data, this is a case when everything changed. So the core measurement changed, water changed, signature changed, everything changed, that's thalamus. What I'm trying to show you, convince you that from what I showed you in the beginning, when we say something is changing, maybe some atrophy, we suddenly have a lot more information. It's still not the molecular level that we would maybe want. And maybe probably we would never get with MRI. But it's much more. We get more specificity for different sources of variability within the tissue. Okay. Now, the last bit will be about, I'm going to be fast. The last bit going to be about theory of aging. And there is these two theories that we find in literature. We find them interesting and wonder if we can say something about them. So one of them is this idea of common cause. So there is a common cause drive aging, and you would see it's implemented or phenotype of that along the brain in different regions. So it's something that drives that's aging. And the other ones, is the idea of the mosaic. Like different brain, the idea here is that different brain aging have a very different perspective or change during aging. It's not a one thing. And different area would show then a very different phenotype of aging. So we said, okay, we can check that. The way we checked it is that it's using our measurement. So we have a measurement of molecular composition, right? We have a measurement of water fraction. We have a measurement of atrophy, just the thing I showed you in the beginning, this like gross change in terms of volume. We have these other measurements which I didn't mention, But MRI is very sensitive to the iron content because it's a magnetic, because of its magnetic features. So we have several different kind of biological measurement, and we ask are they covariant, essentially. And so what I'm putting here is the y-axis is the aging effect size, so how much it changed in terms of age. And this is and the axis is different regions. There was some subdivision along the white matter, subcortical region, and cortex. The gray line here is one of the molecular compositions that I showed you, one of them. There's few. I'm going to show one here. And so you can see it's not the same across the brain. And then I'm going to put the other measurements. And they actually doesn't covary so much. Some of them does. At some level there is covariance, but not so much. And if I'm just going to color each region according to the one which had the biggest effect, for illustration at least, we're going to get this mosaic. And so we think this is a supporting evidence for this thought of the mosaic process of aging. Okay. And so I would like to conclude, actually. I showed you that there is a dependency between quantitative MRI and water fraction, and that creates ambiguity. And if we remove the water effect, that can remove some of this ambiguity. I showed you that, at least in samples, right, when it was purified lipid and we control for everything, we can actually separate between the type of phospholipid. So this measurement is sensitive to the type of phospholipid if we account for water. I showed you that in the human brain, we have unique signatures for different regions when we're doing this analysis. And then I showed you that this multiple dimension signature or slopes per brain region was covaried with histological work of profile of phospholipid and proteins, actually, and gene expression, right? And the last bit was two bits, actually. There was the aging effect that we showed. There was actually a dramatic aging effect in these profiles. And the last thing was that we can use this kind of information or specificity to maybe test or check or validate theories of aging. And this is it. With that, I will thank my group. It is just fun to be with, and I'm grateful for my group. And thank you.